Welcome back to my channel, my dear student. Welcome to my channel. That is Lakshmi Bio tutorial. The topic I'm going to discuss is about gel electrophoresis. Gel electrophoresis is a technique for separation of element, whether it be DNA, protein, or RNA. It is done by applying electric current. First of all, the material we used to separate was allowed to run on a gel slab, on a slab, and the gel used was the agar gel. Mostly commonly, there are many gel, but the mostly which are used is the agar gel. And now we are discussing about DNA. The DNA after isolation, DNA is allowed to run through the machine, and then when in any case, the it has a positive cathode and an anode. The positive one will go toward the cathode, and the negative one will go to the anode. After applying the electric current, and then the material which we used to separate is has been flown, and the one with heavy one will remain at the bottom, and the one which is lighter one will go forward, and then in case of DNA, when we will see some segment at the bottom or at the front, or at the top. But and after that, when we dilute or something, or when we add it with the ethidium bromide solution, we will get the DNA in the we will get the DNA and in a bright orange color, and we will see the DNA with the help of the gel electrophoresis. And then we discuss about the role of the vector in transferring from the organism to the target host. Now there are certain methods which are used for transfer without vector. Some are microinjection, electroporation, gun transfer, etc. In microinjection, the required DNA segment is extracted from the organism, and then it is introduced into the target host, or competent host, or the target organ with the help of a microneedle or a microinjection. In the electroporation, the genes are for to introduce into through the pore, and in gun gene electroporation, the 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 extracted genes are then shoot at a high velocity to the required to the competent host, and then it will take up and it will express the threat, and hence we get the desired product. Now it's this is about the electrophoresis and vectorless gene transfer. Thank you for watching my video.